My name is Scott Friedmann. I'm the Chief of the Division of Liver Diseases at the Icahn School of Medicine at Mount Sinai, as well as the Dean for Therapeutic Discovery at that institution. I'm co-authoring a review of advances in fibrosis or scarring of the liver, which represents a common pathway of liver response to injury. And as we have learned over the years, fibrosis is very important to the development of liver failure, liver cancer, and represents a huge challenge for the field. Virtually all chronic liver diseases that culminate in cirrhosis, which is the last or end stage of chronic liver disease, do so by accumulating scar or fibrosis, or extracellular matrix as it's called. And it turns out that all the complications of chronic liver disease, or virtually all of them, are a direct result of the accumulation of scar. And so it stands to reason that as we begin to make inroads in understanding the basis for scar deposition in liver, we're really uncovering opportunities for antifibrotic therapy that could slow or prevent the progression to end-stage liver disease. It's been very gratifying for me personally and for the field at large to have witnessed a steady and I would say quite impressive accumulation of knowledge in the field of fibrosis. I entered the field as a fellow at UC San Francisco in 1983, at which time we weren't clear about which cells actually make fibrosis or scar in the liver. And I'm proud that one of my contributions was to help clarify the role of a very important cell type known as the hepatic stellate cell by developing a method to isolate the cell, grow it in primary culture, and characterize its ability to make different scar molecules both in culture and then ultimately in vivo. So along with other groups, one from the Netherlands, others from Germany, there was a fundamental new understanding of the cells that make scar in liver dating back to the early 1980s. What we have begun to understand better is what other cell types that are like stellate cells in the liver also contribute to the generation of scar, how they contribute in different disease states, and of course, most importantly, what are the signals that drive that activation of hepatic stellate cells that is so important for the deposition of scar. So starting with the ability to isolate and characterize these cells in culture and in liver, in situ, animal liver, and then human liver, we began to piece together a more coherent understanding of how these cells activate, what are the signals that converge on the cells, what are the receptors expressed on the cell surface that respond to those signals, and what are the pathways within the cells that drive the generation of this highly fibrogenic, proliferative, migratory cell type. And again, the real beauty of this enhanced, or I would say comprehensive now, understanding of stellate cell activation is that it has provided many fertile targets for antifibrotic therapies, which we're beginning to see take flower in the form of new therapeutic trials for patients with chronic liver disease. Having really established a very fundamental understanding of the cells that make scar in liver and having identified so many of those signals, there is a flurry of interest and, I would say, progress in exploiting that new understanding to develop specific targeted therapies that turn off scar generation and that ultimately may prevent progression of liver disease. So, for example, the discovery of a class of receptors on the cell known as tyrosine kinase receptors led to efforts to block these receptors with specific small molecules in animal models of fibrosis that are now translating into new clinical trials. Similarly, new insights into how genes are regulated through epigenetics, transcriptional gene regulation, nuclear transcription factors have also yielded new opportunities for therapies, and these too are being tested in clinical trials. They include not only the tyrosine kinase receptor antagonists, also a whole host of nuclear receptor ligands, molecules that block the generation and or the cross-linking of scar once it's laid down. These are all now playing out in clinical trials throughout the world. And it's really based on this fundamental accrual of information about the biology of stellate cell activation that converts these cells into cell types known as myofibroblasts. It's an amazingly exciting time, and I think the future will begin to yield successes in taking that basic information, having translated it into potential therapies, and ultimately the successes should come in the form of successful clinical trials that lead to new therapies for patients with chronic liver disease. And of course, these aren't being tested in isolation. They're being tested in combination with other therapies that will attack other pathways that converge on fibrosis. So for example, we can envision, and in fact are already seeing in clinical trials, drugs that may not only attack the ability of the liver to generate scar, but also to attack the generation of damage to hepatocytes, blocking inflammation, blocking the accumulation of fat in the liver. All of these ultimately will combine to yield what I believe will be a menu of opportunities and multimodal therapy for patients with chronic liver disease. I would say that the future of fibrosis research is extremely bright because of our successes collectively, and I want to emphasize that this has been a wonderfully collaborative field. The community of investigators in fibrosis have worked together in friendship from the US, Europe, Asia, and other regions of the world to create this new understanding, this heightened opportunity. And so the investments that were made are now beginning to yield important new advances and fruit that will be ripe for the picking in the form of new therapies and new opportunities to treat liver disease.